So a guy named Erickson did an experiment to understand how it is that the nervous system codes different types of flavors. So basically, what he did was he gave different taste stimuli to rats and made recordings from the chorda tympani nerve, this nerve that starts from the tongue and projects into the nucleus of the solitary tract. He identified two substances, ammonium chloride and potassium chloride, two very similar substances. They caused fiber firing, so they caused a pattern of activation in the chorda tympani that was very similar to each other. So it was kind of hard to tell the difference. So whenever you gave a rat ammonium chloride or potassium chloride, and if you just recorded the activity in the chorda tympani nerve, you wouldn't be able to tell which chemical they had. But the salt, sodium chloride, caused a different pattern of firing from these other two. So you could tell the difference between sodium chloride and, say, ammonium chloride or potassium chloride, but you couldn't tell the difference in neural firing between the ammonium chloride or the potassium chloride. They both looked the same, but even though they both looked the same, they were both different from sodium chloride. Then what he did after he figured out what the firing looked like, the rats were then trained by shocking them when they drank potassium chloride. So basically, he made it aversive for them to drink the potassium chloride. So whenever they drank it, they would get a little foot shock, and they would be like, "Oh, I don't want to drink that anymore." So that he scared them off of potassium chloride, right? And then tested them later on, and when they were given a choice, they avoided drinking ammonium chloride, but they continued to drink sodium chloride. Now remember, they were scared off the potassium chloride with foot shocks. Then later on, after they were scared off the potassium chloride, we put them in the cage and let them drink, let them see if they would drink ammonium chloride, and they didn't drink it, which means that perceptually it tasted the same to them, which is good because remember, the firing patterns on the chorda tympani were also the same. So it's really nice that we had that matchup where the same firing patterns seemed to produce the same perception because they were avoiding this drink thinking that it was the same as the one they were scared off of. But they did drink the sodium chloride. They did drink it. They weren't scared off of it. So they were able to distinguish sodium chloride, ammonium chloride, at the perceptual level. We were able to show that rats could distinguish these two chemicals at the perceptual level as well as at the neural level inside of the chorda tympani. So here's a picture here of the firing rates of the different chemicals. We have sodium chloride, which for some reason is just dots. I don't know why there's not a line in this picture, but you can see these little dots. The firing in the first seconds of activity. Each one of these letters represents one fiber. So they found one fiber in the nerve of these rats and then recorded it. And notice that in the fibers, for example, that responded very highly to sodium chloride, they didn't respond at all, or they responded very low to either the potassium chloride or the other compound here, the ammonium chloride. The ammonium chloride. Oops, sorry about that. The ammonium chloride. So remember that the ammonium chloride and the potassium chloride look about the same, which we can see in these pictures. Notice that the red bar and the green bar look pretty. They both look more or less the same. They both start kind of high here and they both go down here, meaning that we have, for any one of these given neurons, we have pretty much the same pattern of firing, whether it's the ammonium chloride or the potassium chloride. However, the salt has a very distinct pattern of firing. Notice that for those fibers, for those cells that are being measured, the ones that only responded to potassium chloride didn't respond very well to the to the other two chemicals, whereas the ones that responded well to the two chemicals, they didn't respond that well at all to the sodium chloride. So this is good, really good data showing this distinction between sodium chloride and the other two chemicals, and we were also able to get it with the perceptions as well, because we knew that they wouldn't drink the stuff that tasted like the stuff that was scary to them. So one of the theories in how taste works has to do with specificity coding. We know now that we have these basic receptors, right? But do we have receptors that specifically code for certain tastes? Do we have those? And there's been some experiments done to try to identify what the mammalian taste system works like. One of the classics is by Mueller. So basically what he did was he created mice, these genetic mice, and what they're referred to as knockout mice. They created a mouse that has a human receptor in it. So they took the gene that produces the human taste receptor for a compound called PTC. And PTC is a big old long name. It's it's in your book, but it's this big old long name that I'm not even going to try to pronounce. But PTC is very bitter. It has a very bitter taste. And humans can perceive that, and we've identified the gene that produces the protein receptor that responds to that chemical in our tongue. So they took that gene out of the human genome, popped it into a mouse genome, and created these mice that were expressing these human receptors for PTC. Now, normal mice don't have these receptors, but when you create a mouse that has the human receptor, it will all of a sudden have it. It was a success. In other words, they were able to create mice that could perceive PTC, because normally mice don't perceive that bitter flavor at all. But the mice that had this gene inserted into their genome did express it. The experiment was a success, but not all of the data show the same results. So let's go into that. So here's some data. If you look at the x-axis going from 0 across, it's a logarithmic scale going across the bottom, you see the concentration of PTC. So you go all the way from hardly any PTC to lots of PTC, going from left to right. No PTC to lots of PTC. And then the other, on the y-axis, we have the percentage change in licking. So how much is the mouse licking the water, the little water bottle that contains a concentration of PTC in it. Now notice the blue line across the top. That is a normal mouse. Notice that normal mice, they don't care. They're going to just keep drinking away the water, even if it has very high concentrations of PTC in it, because they can't taste it, because they don't have the gene for it, and therefore they do not have the receptor cell for it. But notice the mouse with the PTC receptor, the higher the concentration gets of this bitter chemical, the less they drink, the less they lick water from that has this stuff in it. Very clearly indicating that this receptor was doing its thing inside these mice. So here's an interesting the response, right? So in a normal mouse, right? A normal mouse. Let's look in the first part of this table here. A normal mouse does not have a PTC receptor, so it doesn't avoid the PTC. A normal mouse, however, does have a receptor for another bitter chemical called, it's abbreviated as CYX, and that's another chemical with a really long name I'm not going to try to pronounce, but it's just CYX. Normal mice do have CYX receptors, so they do avoid it. They if you do that same experiment that we did here, but with CYX instead of PTC, you will show that decrease the higher the concentration of CYX gets. However, in the cloned mouse, in the mouse that had the PTC receptor, they did have a PTC receptor. And the scientists, Mueller also removed the CYX receptor. So they took out the CYX receptor, or the gene for the CYX receptor, and noticed that it doesn't avoid the CYX anymore like normal mice do. So this is really fairly strong evidence of a direct relationship between a specific receptor that codes for specific flavors represented in the mammalian taste perception. In another experiment by Sato, they recorded 66. They recorded activity from 66 different fibers in the monkey's chorda tympani, and the results show that the fibers, that there were fibers that responded best to one of the basic tastes. So you look at 66 fibers, and out of those 66 fibers, they found some that really responded to sweet. They found others that really responded to salty, others that responded to mostly sour, and still others that responded only to bitter. And when they responded, say, for example, to bitter, they didn't respond very well to the others. So this gives us evidence that there is some really specific coding going on among the fibers, that where there are certain fibers that are specialized for transmitting information about certain tastes to the central nervous system, which gives us some indication that we do have this specific, we do have a bit of specificity coding going on. So here's a picture of these some of these actual data. The darker you see these lines bunched together, the more firing there is. Every time the fiber fires, there's one little spike goes up. You'll notice that, for example, for this neuron here, that's represented in part A of this figure, this neuron would be classified as a sucrose selective neuron. So sucrose is table sugar, basically. So it's sweet. And so we have so this fiber responded very well to sucrose, to table sugar, but it didn't respond to salt, hydrogen chloride, or quinine, or QHCl. Whereas another neuron, B, that's represented in B, responded very well to NaCl, which is sodium chloride, which is table salt. Responded very well to table salt, but hardly at all to sucrose or the other chemicals. And then we have yet another neuron, C, which responded equally to the three salty bitter chemicals, but not too much to the sweet chemical sucrose. So again, this is really good physiological evidence for this specificity coding. In other words, specific structures in our nerves that send information for specific flavors. All right, so in this picture here, we have two different neurons represented. One's is represented in the top of in the top graph, and another neuron is represented in the bottom graph. And each one of these two neurons is exposed to a number of different chemicals. Notice that the chemicals are roughly grouped so that the salty ones are over on the left side of the graph. And in the middle of the graph is bitter-sour chemicals, and then sweet chemicals are at the end of the graph. Now notice the neuron up in the top. It is very responsive to salty flavors. Notice that it goes crazy. That blue dotted line goes really high with the salty chemical. But if you look at the blue dotted line, across the other chemicals, it doesn't respond as highly. Now, if you look at the neuron in B, you'll notice that the blue dotted line, the blue dash line, it goes all over the place. So this neuron in B responds equally well to pretty much everything, salty, bitter, sour, and sweet. Now, the purple line that you see in both of these graphs is the result of giving a sodium channel blocker to putting it on the tongue. So if you remember, salty flavors are perceived when sodium ions go through a sodium channel into the taste cell. So if you block the sodium channel, you would think that, well, you shouldn't be able to taste salt anymore. And you would also surmise that the cells that respond to salt, the fibers that respond to salt, probably shouldn't be firing anymore. Well, that turns out to be the case. If you look at the figure A, notice that this cell, which responds very well to salt, but not to anything else, notice how the salt response goes way down here. Notice it's very different. The purple line is very different from the dash line at the very beginning of this graph. Whereas if you look at the other graph, notice there's no difference between the purple line and the blue dash line. They are both almost the same because that cell doesn't respond to salt, and so therefore we presume it doesn't respond to the closing of or the sodium activation of sodium channels.